The immune system is a complex network of cells, tissues, and organs that work together to defend the body against infections, toxins, and abnormal cells. The fundamental components of this system include various immune cells, which originate from hematopoietic stem cells (HSCs) and specialized organs that support immune cell development and function. Hematopoiesis, origin of immune cells. Hematopoiesis is the process by which all blood cells, including immune cells, are formed. It occurs primarily in the bone marrow, where multipotent hematopoietic stem cells (HSCs) differentiate into two major progenitor lineages. Lymphoid lineage, adaptive and some innate immunity. B cells, responsible for antibody production, humoral immunity. T cells, mediators of cell-mediated immunity. Natural killer NK cells, cytotoxic lymphocytes involved in innate immunity. Myeloid lineage, innate immunity and precursor to red blood cells and platelets. Granulocytes. Neutrophils, first responders to infection, phagocytic. Eosinophils, combat parasitic infections, involved in allergic reactions. Basophils, release histamine, involved in allergic responses. Monocytes, macrophages. Monocytes circulate in the blood and differentiate into macrophages in tissues, acting as professional phagocytes. Dendritic cells, DCs, antigen-presenting cells, APCs, that initiate adaptive immune responses. Mast cells, release histamine and other mediators in allergic reactions. Megakaryocytes, give rise to platelets involved in clotting. Erythrocytes, red blood cells, RBCs, primarily oxygen transport but also play roles in immune signaling. Primary and secondary immune organs. Immune organs are categorized into primary, sites of immune cell development, and secondary, sites of immune activation and response. Primary, central, immune organs, bone marrow. Site of hematopoiesis, generation of all blood cells. B-cell maturation occurs here. Thymus. Site of T-cell maturation and selection. Ensures T-cells develop self-tolerance, prevention of autoimmunity. Secondary, peripheral, immune organs, lymph nodes. Filter lymphatic fluid for antigens. House B and T cells, site of adaptive immune response initiation. Spleen. Filters blood for pathogens and damaged cells. Contains red pulp, RBC clearance, and white pulp, lymphocyte-rich areas. Mucosa-associated lymphoid tissues, MALT. Includes Peyer's patches, gut-associated tonsils, and bronchial-associated lymphoid tissues. Defends mucosal surfaces against pathogens. Other lymphoid tissues. Gut-associated lymphoid tissue, bulk, includes Peyer's patches, appendix, and lymphoid aggregates in the intestines. Bronchus-associated lymphoid tissue, bulk, defends respiratory tract. Functional roles of immune cells. Neutrophils and macrophages, phagocytose pathogens and debris. Dendritic cells, present antigens to naive T cells, initiating adaptive immunity. B cells, differentiate into plasma cells that secrete antibodies. T cells, include cytotoxic T cells, kill infected cells, and helper T cells coordinate immune responses. NK cells, recognize and destroy virally infected and cancerous cells. Practice questions. Multiple choice questions, MCQs. Which of the following immune cells originates from the myeloid lineage? A. B cells. B. T cells. C. Natural killer NK cells. D. Neutrophils. Answer: D. Neutrophils. Which organ is responsible for the maturation of T lymphocytes? A. Bone marrow. B. Spleen. C. Thymus. D. Lymph nodes. Answer: C. Thymus. Which secondary lymphoid organ is primarily responsible for filtering blood? A. Lymph nodes. B. Spleen. C. Bone marrow. D. Peyer's patches. Answer: B. Spleen. Previous year question. Which of the following is not a secondary lymphoid organ? A. Spleen. B. Lymph node. C. Thymus. D. Peyer's patches. Answer: C. Thymus. The thymus is a primary lymphoid organ, where T-cell maturation occurs, while the others are secondary lymphoid organs involved in immune responses. This completes the detailed coverage of cells and organs of the immune system, focusing on hematopoiesis, immune cell lineages, and lymphoid organs essential for immune function. Let me know if you need modifications or the next topic. You said. Regulation of hematopoiesis. ChatGPT said. Regulation of hematopoiesis. Hematopoiesis is a tightly regulated process that ensures the continuous production of blood cells from hematopoietic stem cells, HSCs, in the bone marrow. The regulation of hematopoiesis is crucial for maintaining immune homeostasis, responding to infections, and replenishing blood cell populations. This regulation is controlled by a combination of intrinsic factors, genetic and epigenetic control, extrinsic factors, cytokines and growth factors, and microenvironmental signals, bone marrow niche interactions. 1. Hematopoietic stem cells, HSCs, and their regulation. Hematopoietic stem cells, HSCs, are multipotent cells capable of self-renewal and differentiation into all blood cell lineages. Their regulation occurs through several key mechanisms. A. Transcriptional regulation. Transcription factors play a vital role in determining cell fate by regulating gene expression. Some key transcription factors include RUNX1 essential for HSC formation and differentiation. GATA1 drives erythroid and megakaryocyte differentiation. 2.1 regulates myeloid, monocyte, macrophage, dendritic cell, and lymphoid differentiation. Ikaros and Notch signaling, critical for lymphoid lineage commitment. CEBP, CCAAT, enhancer binding proteins involved in myeloid lineage differentiation. These transcription factors work in a network, ensuring the correct lineage commitment and functional maturation of immune cells. B. Epigenetic regulation. Epigenetic mechanisms such as DNA methylation, histone modifications, and non-coding RNAs control gene accessibility and expression. DNA methylation by DNMTs, DNA methyltransferases, regulates lineage-specific gene activation or suppression. Histone modifications like acetylation and methylation determine chromatin accessibility for transcription factors. MicroRNAs, mRNAs, fine-tune gene expression by targeting mRNA for degradation or translational repression. These epigenetic modifications ensure hematopoiesis is dynamic and adaptable to physiological needs. 2. Extrinsic regulation, cytokines and growth factors. Extrinsic signals, primarily cytokines and growth factors, play an essential role in directing HSC self-renewal and differentiation. The major regulators include stem cell factor SCF, c-KIT ligand supports HSC survival and self-renewal. Interleukins, ILs. IL-3 stimulates myeloid progenitors. IL-7 essential for lymphoid lineage commitment, B and T cells. IL-5 promotes eosinophil differentiation. Granulocyte macrophage colony stimulating factor, GM-CSF, stimulates granulocyte and monocyte differentiation. Granulocyte colony stimulating factor, GCSF, drives neutrophil production. Macrophage colony stimulating factor, MCSF, promotes monocyte macrophage differentiation. Erythropoietin EPO, produced by the kidneys, stimulates erythroid progenitor expansion. Thrombopoietin TPO, essential for megakaryocyte platelet precursor development. These cytokines act via receptor-mediated signaling pathways, often involving JAK-STAT, Janus kinase signal transducer and activator of transcription pathways, MAPK, mitogen-activated protein kinase, and PI3K ACT signaling, to drive lineage-specific differentiation. 3. Bone marrow microenvironment niche regulation. The bone marrow provides a specialized microenvironment, or niche, that supports HSC maintenance and differentiation. It consists of osteoblastic endosteal niche, located near the bone surface, maintains HSC quiescence dormancy. Vascular niche, associated with blood vessels, supports HSC activation and mobilization. Key components of the niche include mesenchymal stromal cells MSCs provide support via cytokines like SCF and CXCL12. Osteoblasts regulate HSC maintenance. Endothelial cells promote HSC proliferation and migration. Sympathetic nervous system SNS modulates HSC mobilization via neurotransmitter release. The interaction of HSCs with niche components through adhesion molecules like integrins BLA4, VCAM1 cadherins, and CXCR4 CXCL12 signaling is crucial for hematopoietic regulation. 4. Feedback regulation and homeostasis. Hematopoiesis is adjusted based on the body's needs. Several feedback mechanisms help maintain balance. Negative feedback via cytokines. High neutrophil counts suppress GCSF production, reducing neutrophil differentiation. EPO production increases in response to hypoxia, stimulating red blood cell production. Apoptosis of mature cells prevents excess immune cell accumulation. Inflammatory signals e.g., TNF-alpha, IFN-gamma can enhance or suppress specific hematopoietic pathways depending on context e.g., chronic inflammation suppresses HSC function. 5. Clinical significance of hematopoietic regulation. Dysregulation of hematopoiesis leads to hematological disorders. Leukemia, uncontrolled proliferation of hematopoietic progenitors. Aplastic anemia, failure of hematopoiesis due to bone marrow suppression. Myelodysplastic syndromes MDS, abnormal differentiation leading to ineffective hematopoiesis. Cytokine therapies, recombinant EPO for anemia and GCSF for neutropenia are used clinically. Practice questions. Multiple choice questions MCQs. Which of the following transcription factors is essential for myeloid differentiation? A-GATA-1. B2.1C, RUNX1D, Icaros. Answer, B2.1. Which cytokine is primarily responsible for erythropoiesis? A. IL-3. B. GCSF. C. Erythropoietin EPO, D. Thrombopoietin TPO. Answer, C. Erythropoietin EPO. Which of the following cells is a key component of the hematopoietic niche in the bone marrow? A. Osteoblasts. B. Neutrophils. C. Erythrocytes. D. Basophils. Answer, A. Osteoblasts. Previous year question. Which signaling pathway is most commonly associated with hematopoietic cytokine action? A. WNT signaling. B. Hedgehog signaling. C. JAK-STAT pathway. D. TGF-beta pathway. Answer, C. JAK-STAT pathway. Cytokines like IL-3, EPO, and GCSF primarily act via the JAK-STAT signaling pathway. This concludes the detailed regulation of hematopoiesis covering intrinsic factors, extrinsic signals, microenvironmental influences, and feedback control. Let me know if you need any modifications or the next topic. You said. Development of T cells and B cells. ChatGPT said. Development of T cells and B cells. T and B lymphocytes are the key players of the adaptive immune system. Their development involves multiple stages of differentiation, selection, and maturation to ensure proper immune function while preventing autoimmunity. T cell development occurs in the thymus, while B cells mature in the bone marrow. 1. T cell development. T cell development occurs in the thymus, where progenitor cells from the bone marrow migrate and undergo a highly regulated process involving differentiation, selection, and commitment to specific T cell lineages. Stages of T cell development: hematopoietic stem cell (HSC) stage. HSCs originate in the bone marrow and migrate to the thymus. They express CD34, a stem cell marker, and do not yet have T cell characteristics. Double negative DN stage CD4-CD8. At this stage, cells lack CD4 and CD8 markers. Further divided into DN1, DN2, DN3, and DN4 stages, marked by rearrangement of T cell receptor TCR genes. TCR rearrangement begins with the beta chain, which pairs with a surrogate alpha chain pre-TCR for signaling. Checkpoint 1. Only cells with a functional pre-TCR proceed to the next stage. Double positive DP stage CD4-CD8. Cells express both CD4 and CD8 co-receptors. TCR alpha chain rearrangement occurs, allowing a fully functional TCR AB complex to be expressed. Positive selection. Thymic epithelial cells present self-MHC molecules to developing T cells. Cells that recognize self-MHC weakly survive. Cells that fail to recognize MHC undergo apoptosis. Single positive SP stage CD4 or CD8. Cells commit to either CD4 helper T cells MHC2 restricted or CD8 cytotoxic T cells MHCI restricted. Negative selection. T cells that bind too strongly to self-MHC undergo apoptosis to prevent autoimmunity. Mature naive T cells. Surviving T cells exit the thymus and circulate in the periphery. They remain naive until encountering their specific antigen in lymphoid tissues. Regulation of T cell development. Transcription factors. Notch1 essential for T cell lineage commitment. TBET drives Th1 differentiation. GATA3 promotes Th2 differentiation. FOXP3 regulates T regulatory TREG cell development. Cytokines and signals. IL-7 critical for early T cell survival and expansion. TCR signaling strength determines survival during selection. T cell selection and tolerance. Positive selection cortex of thymus ensures cells recognize self-MHC. Negative selection medulla of thymus eliminates self-reactive T cells to prevent autoimmunity. Outcome. Weak binding to self-MHC right pointing arrow survival, no binding right pointing arrow apoptosis. Strong binding right pointing arrow apoptosis. Negative selection 2. B cell development. B cell development occurs in the bone marrow, followed by activation and maturation in peripheral lymphoid organs. The process involves gene rearrangement, selection, and functional differentiation. Stages of B cell development: hematopoietic stem cell (HSC) stage. HSCs in the bone marrow give rise to common lymphoid progenitors, CLPs, that can differentiate into B or T cells. Pro-B cell stage. Heavy chain IgH gene rearrangement begins with the DJ recombination followed by VDJ recombination. Expresses CD19 and CD79A, early B cell markers. Pre-B cell stage. Heavy chain successfully synthesized and paired with a surrogate light chain, pre-BCR complex. Checkpoint 1, cells with functional pre-BCR proliferate. Light chain rearrangement, IgL, begins. Immature B cell stage. Expresses functional IgM on the surface. Negative selection occurs. Strong self-antigen recognition, right pointing arrow, apoptosis or receptor editing to prevent autoimmunity. Mature naive B cell stage. Expresses both IgM and IgD. Migrates to secondary lymphoid organs, lymph nodes, spleen, MALT for activation. Activation and differentiation of B cells, T-dependent activation, most common. Requires help from CD4 T cells via MHC2 and co-stimulation. Leads to class switching, IgM right pointing arrow, IG, IgA, or IgE, somatic hypermutation, and memory B cell formation. T-independent activation. Triggered by highly repetitive antigens like bacterial polysaccharides. Produces only IgM with no memory response. Plasma cell differentiation. B cells differentiate into antibody-secreting plasma cells or memory B cells for long-term immunity. Regulation of B cell development. Transcription factors. PAX5 essential for B cell lineage commitment. E2A, EBF1 regulate early B cell differentiation. Cytokines. IL-7 supports B cell proliferation. BAFF B cell activating factor essential for B cell survival in the periphery. Practice questions. Multiple choice questions, MCQs. Which of the following is the first checkpoint in T cell development? A, positive selection in the thymus. B, pre-TCR signaling at the DN stage. C, negative selection in the thymus. D, CD4, CD8 lineage commitment. Answer, B, pre-TCR signaling at the DN stage. What is the function of AIR, autoimmune regulator, in the thymus? A, promotes positive selection. B, stimulates B cell development. C, aids in antigen presentation. D, induces tolerance by presenting self-antigens. Answer, D, induces tolerance by presenting self-antigens. Which immunoglobulin is first expressed on immature B cells? A, IgA. B, IgC, IgM. D, IgE. Answer, C, IgM. Previous year question. Which of the following best describes the outcome of strong self-antigen recognition during B cell development? A, positive selection. B, activation and class switching. C, apoptosis or receptor editing. D, differentiation into memory B cells. Answer. C, apoptosis or receptor editing. Strong recognition of self-antigens leads to negative selection or receptor editing to prevent autoimmunity. This concludes the detailed development of T cells and B cells, covering their differentiation, selection, and regulatory mechanisms. Let me know if you need modifications or the next topic. You said. Primary, secondary, and tertiary lymphoid organs, ChatGPT said. Primary, secondary, and tertiary lymphoid organs. The immune system is organized into lymphoid organs, which play distinct roles in immune cell development, activation, and response. These organs are classified into primary, central, secondary, peripheral, and tertiary lymphoid organs based on their functions. 1. Primary, central, lymphoid organs. Primary lymphoid organs are responsible for the generation, maturation, and selection of immune cells, lymphocytes. These include a, bone marrow. Site of hematopoiesis, blood cell production, including the development of B cells, T cell precursors, and other immune cells. B cell maturation occurs here, including immunoglobulin gene rearrangement, VDJ recombination. Negative selection, self-tolerance mechanism, to eliminate self-reactive B cells. Mature naive B cells express IgM and IgD and migrate to secondary lymphoid organs. T cell precursors migrate from the bone marrow to the thymus for further development. B, thymus. Site of T cell maturation and differentiation. T cell selection process. Positive selection in the thymic cortex ensures T cells recognize self-MHC molecules. Negative selection in the medulla removes T cells with high affinity for self-antigens to prevent autoimmunity. The thymus is most active during childhood and shrinks involutes with age. Functions of primary lymphoid organs. Generation of lymphocytes from hematopoietic stem cells, HSCs. Education and selection of immune cells to ensure self-tolerance. Export of naive, functional T and B cells to secondary lymphoid organs. 2. Secondary peripheral lymphoid organs. Secondary lymphoid organs are the sites where mature lymphocytes encounter antigens, become activated, proliferate, and differentiate into effector cells. These include a, lymph nodes. Small, bean-shaped structures located along lymphatic vessels. Function. Filter lymph and trap antigens from tissues. Provide an environment for T and B cell activation. Dendritic cells, DCs, present antigens to naive T cells. Structure. Cortex, contains B cell follicles and germinal centers, where B cells proliferate and undergo somatic hypermutation. Paracortex, rich in T cells and dendritic cells. Medulla, contains plasma cells producing antibodies. B, spleen. Filters blood and detects bloodborne pathogens. Structure. White pulp, contains T cells, periarterial or lymphoid sheath, pals, and B cell follicles. Red pulp, removes aged RBCs and recycles iron. Function. Initiates immune responses against systemic infections. Removes old, damaged RBCs via macrophages. C. Mucosa-associated lymphoid tissue, MALT, found in mucosal surfaces, gut, respiratory tract, urogenital tract. Examples. Gut-associated lymphoid tissue, GALT, includes Peyer's patches, appendix, tonsils. Bronchus-associated lymphoid tissue, BALT, found in the lungs. Function. Provides localized immune surveillance against pathogens entering through mucosal surfaces. D. Other secondary lymphoid tissues. Tonsils and adenoids, protect against respiratory infections. Peyer's patches, found in the intestine, involved in gut immunity. Functions of secondary lymphoid organs, trap and present antigens to immune cells. Facilitate the interaction between antigen-presenting cells, APCs, and lymphocytes. Support clonal expansion and differentiation of activated lymphocytes. 3. Tertiary lymphoid organs, TLOs. Tertiary lymphoid organs are non-classical immune structures that form in response to chronic inflammation, infections, or autoimmune diseases. These are not present under normal conditions but develop as ectopic lymphoid tissues at sites of persistent immune activation. Characteristics of tertiary lymphoid organs. Resemble lymph nodes but are formed at sites of chronic inflammation, infection, or cancer. Contain B cell follicles, T cells, dendritic cells, and high endothelial venules, HEVs, for lymphocyte trafficking. Examples include rheumatoid arthritis, TLOs, and synovial joints, Hashimoto's thyroiditis, TLOs, in thyroid tissue, chronic infections, e.g., tuberculosis, granulomas, solid tumors, TLOs, near cancerous tissues, functions of tertiary lymphoid organs, enhance local immune responses against infections or tumors, sustain immune activation in chronic inflammatory diseases, facilitate immune cell recruitment and antigen presentation, comparison of lymphoid organs. Feature primary lymphoid organs, secondary lymphoid organs, tertiary lymphoid organs, function, lymphocyte development and selection, lymphocyte activation and immune response, ectopic immune response and inflammation. Examples: bone marrow, thymus, lymph nodes, spleen, MALT, TLOs, and inflamed or diseased tissues. Present at birth, develop under pathological conditions. Antigen exposure, no direct exposure, high exposure to antigens, high exposure to persistent antigens. Practice questions. Multiple choice questions, MCQs. Which of the following is a primary lymphoid organ? A. Lymph node. B. Spleen. C. Thymus. D. Peyer's patches. Answer: C. Thymus. Which of the following structures is responsible for filtering blood and removing old red blood cells? A. Lymph node. B. Spleen. C. Bone marrow. D. Thymus. Answer: B. Spleen. Which type of lymphoid organ is formed at sites of chronic inflammation? A. Primary lymphoid organ. B. Secondary lymphoid organ. C. Tertiary lymphoid organ. D. Bone marrow. Answer: C. Tertiary lymphoid organ. Previous year question. Which of the following statements about MALT is correct? A. It is a primary lymphoid organ. B. It is involved in filtering blood. C. It includes Peyer's patches and tonsils. D. It does not contain lymphocytes. Answer: C. It includes Peyer's patches and tonsils. MALT is a secondary lymphoid organ that protects mucosal surfaces. This completes the detailed primary, secondary, and tertiary lymphoid organ section. Let me know if you need modifications or the next topic. You said. Activation and differentiation of B and T cells. ChatGPT said. Activation and differentiation of B and T cells. The activation and differentiation of B and T cells are essential processes in adaptive immunity, enabling the body to recognize and eliminate pathogens effectively. These processes occur in secondary lymphoid organs, lymph nodes, spleen, MALT, after antigen encounter and involve a series of signaling events that lead to effector and memory cell formation. 1. Activation and differentiation of T cells. T cells require antigen presentation by antigen-presenting cells, APCs, to become activated. This process occurs in lymph nodes or spleen, where dendritic cells, macrophages, and B cells present antigens to naive T cells. Steps of T cell activation: antigen presentation, signal 1. Dendritic cells, DCs, process and present antigens via MHC molecules. CD4 T cells recognize antigen on MHC2, while CD8 T cells recognize antigen on MHCI. The T cell receptor, TCR, binds to the MHC peptide complex, initiating activation. Co-stimulatory signals, signal 2. Co-stimulatory molecules are required for full activation. CD28 T cell binds to B7 APC, providing essential activation signals. Absence of co-stimulation leads to T cell energy, non-responsiveness. Cytokine signaling, signal 3. Differentiation. APC secrete cytokines that direct T cell differentiation into specific subsets. Different cytokines promote different T cell fates. Differentiation of CD4 helper T cells, TH cells. CD4 T cells differentiate into various subsets based on cytokine signals. T cell subset cytokine required function. Th1 cells IL-12, IFN gamma activate macrophages, promote cell-mediated immunity, fight intracellular pathogens. Th2 cells IL-4 promote B-cell activation, antibody production, fight extracellular parasites. Th17 cells IL-6, IL-23, TGF-beta enhance neutrophil response, fight fungal and bacterial infections. Treg cells IL-10, TGF-beta suppress immune responses, maintain tolerance. Differentiation of CD8 cytotoxic T cells, CTLs. Activated CD8 T cells differentiate into cytotoxic T lymphocytes, CTLs, which kill virus-infected and cancerous cells. CTLs use perforin and granzymes to induce apoptosis in target cells. Memory T cells. Some activated T cells become memory T cells, providing long-term immunity. Memory T cells respond faster and more robustly upon re-exposure to the same antigen. 2. Activation and differentiation of B cells. B cell activation occurs in lymph nodes, spleen, and MALT, leading to antibody production and memory B cell formation. Types of B cell activation: T-dependent B cell activation, most common. Requires CD4 T cell help. Leads to strong, long-lasting immune responses with memory formation. T-independent B cell activation occurs without T cell help, primarily against polysaccharide antigens, e.g., bacterial capsules. Produces only IgM, with no memory formation. Steps of T-dependent B cell activation: antigen binding to BCR, signal 1. The B cell receptor, BCR, binds to the antigen. Antigen is internalized, processed, and presented on MHC2. T cell help, signal 2. Activated Th cells recognize antigen on MHC2 of B cells. CD40 B cell binds to CD40L T cell, providing co-stimulation. Cytokine signaling, signal 3. Differentiation. Helper T cells secrete IL-4, IL-5, IL-6, promoting B cell proliferation and differentiation. Differentiation of B cells. Plasma cells secrete large amounts of antibodies, Ig, IgA, IgE. Provide humoral immunity against extracellular pathogens. Memory B cells persist for years, allowing rapid responses to reinfection. Germinal center reaction, affinity maturation and class switching. Occurs in lymph node follicles. Somatic hypermutation, generates high affinity antibodies. Class switching, IgM right pointing arrow Ig, IgA, or IgE depends on cytokine signals. Comparison of B cell and T cell activation feature: T cell activation, B cell activation site, lymph nodes, spleen antigen recognition, MHC bound antigen free floating antigen co-stimulation required. Yes, CD28B7, CD40, yes, CD40, effector cells, helper T cells, cytotoxic T cells, plasma cells, memory formation, yes. Practice questions, multiple choice questions, MCQs. Which of the following is required for full T cell activation? A. MHC recognition alone. B. Antigen presentation and cytokine signal. C. TCR binding alone. D. Perforin and granzyme release. Answer: B. Antigen presentation and cytokine signals. Which cytokine is essential for Th1 differentiation? A. IL-4. BIL-12C IL-10D TGF beta. Answer, BIL-12. Which immunoglobulin is produced first during a primary immune response? A. IgA. B. IgC IgMD IgE. Answer, C. IgM. Previous year question. Which of the following best describes the role of CD40-CD40L interaction? A. It helps in antigen presentation by dendritic cells. B. It is essential for B-cell activation and class switching. C. It suppresses T-cell activation. D. It promotes apoptosis in infected cells. Answer, B. It is essential for B-cell activation and class switching. CD40-CD40L interaction is crucial for B-cell proliferation and Ig class switching. This concludes the activation and differentiation of B and T cells section. Let me know if you need modifications or the next topic. You said, B and T cell receptors. ChatGPT said, B and T cell receptors BCR and TCR. The B cell receptor BCR and T cell receptor TCR are essential for adaptive immunity, allowing B and T cells to recognize and respond to specific antigens. While both receptors are involved in antigen recognition, they differ in structure, antigen recognition mechanisms, and signaling pathways. 1. B cell receptor BCR, structure of BCR. The BCR is a membrane-bound immunoglobulin antibody associated with signaling proteins. Composed of 2 heavy chains H chains and 2 light chains L chains forming a Y-shaped structure. Variable B regions, responsible for antigen binding, Fab region. Constant C regions, determine antibody class IgM, Ig, IgA, IgE, IgD. Signaling complex Ig alpha and Ig beta, also called CD79 and CD79B, transmits activation signals. Antigen recognition by BCR. BCRs can bind directly to native unprocessed antigens in a free form. Recognize proteins, carbohydrates, lipids, and nucleic acids. Each B cell has a unique BCR due to VD-J recombination, which generates antigen receptor diversity. BCR activation and signaling. Antigen binding. The antigen binds to the BCR, leading to receptor clustering. Signaling via Ig alpha, Ig beta, CD79A, CD79B activates kinases LIN, SYK, BTK. Downstream activation leads to calcium influx, activation of transcription factors NF-kappa B, AP1, and B cell differentiation. Outcome: activated B cells differentiate into plasma cells, antibody secretion, or memory B cells. BCR functions. Initiates B cell activation upon antigen binding. Triggers antibody production after differentiation into plasma cells. Acts as an antigen-presenting receptor for T cell help via MHC2. Induces class switching and affinity maturation in germinal centers. 2. T cell receptor TCR. Structure of TCR. The TCR is a membrane-bound heterodimer composed of 2 chains, alpha and beta chains most common, Ab T cells, OR gamma and delta chains, grand T cells, less common but important in mucosal immunity. Contains variable B regions, recognize antigen MHC complexes. Constant C regions, provide structural stability. Transmembrane region, anchors TCR to the membrane. CD3 complex epsilon, delta, gamma, and zeta chains essential for signal transduction. Antigen recognition by TCR. TCRs only recognize peptide antigens presented on MHC molecules. CD4 T cells recognize MHC2 on APCs. CD8 T cells recognize MHCI on all nucleated cells. Unlike BCRs, TCRs cannot bind free-floating antigens and require antigen processing. TCR activation and signaling. Antigen presentation. Dendritic cells, macrophages, or B cells present processed peptides on MHC. TCR binding to MHC peptide complex, signal 1, the TCR binds to the antigen MHC complex on APCs. Co-stimulation, signal 2. CD28 T cell binds to B7 APC, ensuring full activation. Lack of co-stimulation leads to anergy, non-responsiveness. Cytokine signaling, signal 3, APCs release cytokines, guiding T cell differentiation. Outcome: activated T cells proliferate and differentiate into effector T cells Th1, Th2, Th17, TREG, CTLs, or memory T cells. TCR functions. Initiates T cell activation upon antigen MHC binding. Directs immune responses by differentiating into helper or cytotoxic T cells. Essential for immune memory and long-term protection. 3. Differences between BCR and TCR feature B cell receptor BCR T cell receptor TCR structure antibody-like heavy and light chains Ab or grand heterodimer antigen recognition binds free-floating native antigens recognizes processed peptides on MHC associated signaling complex Ig alpha Ig beta CD79A CD79B CD3 epsilon delta gamma zeta chains co-receptors none but requires CD40 for activation CD4 for MHC2 or CD8 for MHCI antigen type recognized proteins lipids carbohydrates nucleic acids only peptides location of activation lymph nodes. Spleen lymph nodes spleen effector function differentiates into plasma cells secreting antibodies differentiates into helper or cytotoxic T cells memory cell formation. Yes. Practice questions multiple choice questions MCQs. Which of the following molecules is part of the TCR signaling complex? A. CD79A. B. Ig alpha C CD3D MHCI. Answer: C CD3. Which statement about BCR and TCR is correct? A. Both recognize free-floating antigens. B. Both require MHC molecules for antigen recognition. C. BCR binds to native antigens, while TCR binds to peptide MHC complexes. D. BCR and TCR are both secreted into the bloodstream. Answer: C. BCR binds to native antigens, while TCR binds to peptide MHC complexes. Which immune receptor directly recognizes lipid and carbohydrate antigens? A. TCR. B. BCRC CD4D CD3. Answer: B. BCR. Previous year question. What is the role of CD3 in TCR signaling? A. It acts as a co-receptor for antigen binding. B. It stabilizes the MHC antigen complex. C. It transduces activation signals into the T cell. D. It presents antigen to the TCR. Answer: C. It transduces activation signals into the T cell. CD3 is essential for TCR-mediated signal transduction. This completes the B and T cell receptors section. Let me know if you need modifications or the next topic. You said, innate and adaptive immune system. ChatGPT said, innate and adaptive immune system. The immune system is the body's defense mechanism against infections, diseases, and foreign invaders. It is divided into two main components. Innate immune system, nonspecific, first line of defense, adaptive immune system, specific, long-lasting immunity. These systems work together to protect the body from pathogens while maintaining self-tolerance. 1. Innate immune system. The innate immune system provides immediate, nonspecific defense against infections. It does not adapt or remember past infections. Key features of innate immunity present from birth: rapid response, minutes to hours, nonspecific, same response to all pathogens, no memory formation. Acts as the first line of defense. Components of the innate immune system: of physical and chemical barriers. First line of defense, skin, prevents pathogen entry, has antimicrobial peptides. Mucous membranes, trap pathogens in mucous respiratory, gastrointestinal tracts. Cilia, sweep pathogens away from respiratory tract. Enzymes, e.g., lysozyme in saliva, tears, sweat, break bacterial cell walls. Stomach acid, HCl, destroys ingested pathogens. B cellular components, second line of defense, phagocytes, engulf and destroy microbes. Macrophages, reside in tissues, phagocytose microbes. Neutrophils, first responders in infection, kill bacteria via phagocytosis. Dendritic cells, present antigens to adaptive immune cells. Natural killer NK cells, kill virus-infected and cancer cells via perforin and granzymes. Mast cells and basophils, release histamine, cause inflammation. Eosinophils, combat parasitic infections and allergies. C-soluble factors, chemical mediators, complement system. A group of proteins, C1, C9, that enhance phagocytosis, inflammation, and pathogen lysis. Forms membrane attack complex, MAC, to lyse bacteria. Cytokines, cell signaling molecules. Interferons, IFN-alpha, IFN-beta, IFN-gamma, inhibit viral replication. Interleukins, IL-1, IL-6, TNF-alpha, promote inflammation, fever. Inflammatory response. Vasodilation, increased blood flow, and leukocyte recruitment to infection sites. 2. Adaptive immune system. The adaptive immune system provides specific, long-lasting immunity. It develops after exposure to pathogens or vaccines and has memory for future infections. Key features of adaptive immunity: specificity, recognizes unique antigens. Slower response, days to weeks for first exposure. Memory formation, faster response in subsequent exposures. Diverse repertoire of antigen receptors, TCRs and BCRs. Components of adaptive immunity: a humoral immunity, B-cell mediated by B cells and antibodies. B cells recognize extracellular pathogens, bacteria, viruses in blood. Antibody functions. Neutralization, block pathogen entry. Opsonization, mark pathogens for phagocytosis. Complement activation, enhance pathogen destruction. B-cell mediated immunity, T-cell mediated by T cells, CD4 and CD8 T cells. Effective against intracellular pathogens, viruses, some bacteria, cancer cells. Types of T cells. CD4 helper T cells, activate B cells, macrophages, and cytotoxic T cells. CD8 cytotoxic T cells, kill virus-infected and cancer cells via perforin and granzymes. Regulatory T cells, TREGs, suppress immune responses to prevent autoimmunity. 3. Differences between innate and adaptive immunity. Feature: innate immunity, adaptive immunity response time immediate, minutes to hours, delayed, days to weeks, specificity non-specific, highly specific, memory no memory, long-term memory. Main cells: macrophages, neutrophils, dendritic cells, NK cells, B cells, T cells. Mode of action: phagocytosis, inflammation, complement activation, antibodies, T cell mediated killing response to repeat infection, same each time, faster and stronger upon re-exposure. 4. Interaction between innate and adaptive immunity. Innate immunity activates adaptive immunity by presenting antigens to lymphocytes. Dendritic cells bridge innate and adaptive immunity by presenting antigens to T cells and lymph nodes. Cytokines from innate immune cells influence adaptive immune responses. Complement proteins enhance antibody function, linking the two systems. Practice questions. Multiple choice questions, MCQs. Which of the following is a key feature of the innate immune system? A. Memory response. B. Antibody production. C. Rapid, non-specific defense. D. Clonal expansion. Answer: C. Rapid, non-specific defense. Which immune cells act as a bridge between innate and adaptive immunity? A. Neutrophils. B. Dendritic cells. C. NK cells. D. Eosinophils. Answer: B. Dendritic cells. Which type of immunity is primarily responsible for protection against intracellular pathogens? A. Humoral immunity. B. Cell-mediated immunity. C. Complement system. D. Passive immunity. Answer: B. Cell-mediated immunity. Previous year question. Which of the following statements correctly describes the difference between innate and adaptive immunity? A. Innate immunity provides long-term protection, while adaptive immunity acts immediately. B. Adaptive immunity is nonspecific, while innate immunity is highly specific. C. Innate immunity lacks memory, while adaptive immunity generates memory responses. D. Adaptive immunity does not involve T and B cells, while innate immunity does. Answer: C. Innate immunity lacks memory, while adaptive immunity generates memory responses. This concludes the innate and adaptive immune system section. Let me know if you need modifications or the next topic. You said. Antigens, antigenicity and immunogenicity. ChatGPT said. Antigens, antigenicity, and immunogenicity. The immune system recognizes and responds to antigens, which are molecules that trigger an immune response. The ability of an antigen to stimulate the immune system is described by two key properties, antigenicity and immunogenicity. 1. Antigen's definition of antigen. An antigen is any foreign molecule, proteins, polysaccharides, lipids, nucleic acids that can be recognized by B cell receptors, BCRs, T cell receptors, TCRs, or antibodies. Types of antigens are based on source. Exogenous antigens enter from outside: bacteria, viruses, toxins, pollen. Endogenous antigens, generated inside the body, viral proteins in infected cells, tumor antigens. Autoantigens, normally present in the body but mistakenly attacked in autoimmune diseases. Allergens, antigens that trigger allergic reactions, pollen, dust mites, food proteins. B based on immune response. T-dependent antigens, require T cell help for antibody production, proteins. T-independent antigens, directly stimulate B cells without T cell help, polysaccharides, LPS. C based on immunogenicity. Complete antigens, can both bind immune receptors and elicit an immune response. Haptens, small molecules that bind immune receptors but need a carrier protein to be immunogenic, e.g., penicillin, urushiol from poison ivy. 2. Antigenicity. Definition of antigenicity. Antigenicity is the ability of a substance to bind specifically to antibodies or TCRs. It determines how well an antigen interacts with the immune system but does not necessarily induce an immune response. Factors affecting antigenicity. Molecular size. Larger molecules, proteins, polysaccharides, are more antigenic than small molecules. Complexity and heterogeneity. Complex structures with multiple epitopes, proteins, glycoproteins, are more antigenic. Foreignness. Highly foreign molecules elicit stronger immune responses. Degradability. Antigens that can be processed by antigen-presenting cells, APCs, are more antigenic. Route of entry. Different routes, injection, ingestion, inhalation, affect antigen recognition. Epitopes, antigenic determinants. The specific region of an antigen that binds to an antibody or TCR. B-cell epitopes, recognize linear or conformational epitopes. T-cell epitopes, recognize only linear peptide fragments presented on MHC molecules. 3. Immunogenicity. Definition of immunogenicity. Immunogenicity is the ability of an antigen to induce an immune response, T-cell activation, antibody production. Factors affecting immunogenicity. Molecular size. Large molecules and GT, 10 kDa, are more immunogenic. Chemical complexity. More complex structures, proteins, glycoproteins, elicit stronger responses. Foreignness. Greater differences from self-proteins increase immunogenicity. Degradability. Antigens that can be processed and presented by APCs are more immunogenic. Dose and route of administration. Optimal dose and correct administration route, subcutaneous, intramuscular, oral, enhance immune response. Adjuvants. Substances added to vaccines, e.g., aluminum salts, to enhance immunogenicity. Host factors, genetic background, immune status, and previous exposure effect response. Comparison of antigenicity and immunogenicity. Property, antigenicity, immunogenicity. Definition. Ability to bind to immune receptors, BCR, TCR, antibodies. Ability to induce an immune response requires immune activation. No, yes. Haptens. Antigenic but not immunogenic, require a carrier to be immunogenic. Example: carbohydrates, lipids, small peptides, proteins, large polysaccharides. 4. Haptens and carrier proteins. Haptens are small molecules that can bind antibodies but cannot trigger an immune response alone. They need to be attached to a carrier protein, albumin, hemocyanin, to become immunogenic. Examples of haptens. Penicillin can cause allergic reactions when bound to serum proteins. Urushiol, poison ivy, causes skin hypersensitivity. 5. Superantigens. Superantigens are special antigens that cause excessive immune activation by directly binding TCRs and MHC molecules, bypassing normal antigen processing. Cause massive cytokine release, leading to toxic shock syndrome, TSS. Examples. Staphylococcal enterotoxin, SE, toxic shock syndrome, toxin 1, TSST1, practice questions, multiple choice questions, MCQs. Which of the following is a characteristic of an antigen but not necessarily an immunogen? A. Can bind to immune receptors. B. Can stimulate an immune response. C. Requires a carrier protein. D. Induces memory formation. Answer. A. Can bind to immune receptors. Which of the following factors increases an antigen?